ASYIRAH, have you ever wondered how vaccines stay stable without refrigeration and how frozen food keeps its texture when defrosted?
It's a really good question, Amira, and the answer might surprise you. It all comes down to a simple sugar called trehalose. And today, we are going to explore why this molecule is revolutionizing both biotechnology and pharmaceutical preservation. Let's go.
So what is actually trehalose? Trehalose is a natural occurring disaccharide composed of two glucose molecules linked by an alpha-alpha-1,1 glycosidic bond. It is commonly found in plants, fungi, insect, yeast, and various microorganisms. Trehalose is well known for its ability to protect cell and biomolecules from environmental stress, like helping organisms survive in extreme conditions. Due to these unique protective properties, trehalose is widely used in the food, pharmaceutical, biotechnology, cosmetic, and medical industries. To the next part, which this slide will show you the structure of trehalose in different form. At the top, we can see the 2D structure and the 3D structure, which will help us to understand how the atoms are arranged. While at the bottom, we can see the Haworth and the Fischer projection, which are the two common ways to represent sugar molecule. As I said, trehalose is made of two glucose molecules joined by an alpha-alpha-1,1 glycosidic bond, which making it a unique and stable disaccharide. Now that you know a bit of trehalose and its structure, next I will explain about the chemical and the physical properties of trehalose. Trehalose has the molecular formula of C12H22, O11 and molecular mass of 342.30 grams per mole. It appears as a white crystalline powder and melts around 97 to 99 degrees Celsius. It is a non-reducing disaccharide, which means it doesn't show reducing properties. Trehalose is also highly stable under heat, acidic, and alkaline condition. Finally, the enzyme trehalase can break trehalose down into two glucose molecules.
So, that's a bit about what trehalose is and its properties. But have you ever wondered where does actually trehalose come from? Like, how does the body or a plant even make it?
Actually, that sounds really interesting. You know, trehalose isn't just made in lab. In living organisms, actually synthesize it naturally through a specific pathway. So, for example, bacteria, fungi, even plants, they all produce trehalose on their own.
Wait, really? So there's like a whole biological process behind it.
Exactly, and it's actually a two-step process. Let me walk you through how it works.
So how is trehalose actually produced inside living cells? To conclude, 5 distinct enzymatic pathways have been identified for trehalose biosynthesis. However, the most common and best studied route is the OTSAP pathway, also known as the TPS or TPP pathway. This pathway works in 2 straightforward steps. So the first step is the enzyme OTSA or TPS1 in yeast combines glucose 6-phosphate and UDP glucose to form an intermediate compound called trehalose 6-phosphate or T6B. Step 2, the enzyme OTSB or TPS2 then remove the phosphate group from T6B, releasing free trehalose and inorganic phosphate. One crucial point, T6B is not merely a precursor, it also serves as a critical signaling molecule that helps regulate carbon metabolism in the cell. So now let us look at what makes the structure of trehalose so chemically special, and that is an alpha-alpha-1,1 glycosidic bond. Both glucose units in trehalose are joined at their anomeric carbon 1 positions, both in the alpha configuration forming an alpha-alpha-1,1 linkage. This creates a non-reducing disaccharide, meaning there is no free aldehyde or ketone group at either end of the molecule. As a result, trehalose does not undergo the Maillard-Brownian reaction, unlike sucrose or glucose. It also shows exceptional chemical stability under high temperatures and acidic conditions. Now, let us explore the biological functions of trehalose and how it protects organisms from extreme environmental stress. So, trehalose plays 4 major roles inside cells. The first one, energy reserve and carbon source, released during starvation or metabolic stress. Second, osmoprotectant, helps cells regulate water balance under osmotic pressure. Third, chemical chaperone, it assists proteins in maintaining their correct 3-dimensional shape. Last, free radical scavenger, it neutralizes reactive oxygen species, ROS. So, one of the most scientifically studied roles of trehalose is the ability to induce autophagy. The cell's own internal cleaning process where damaged proteins and organelles are broken down and recycled. Trehalose promotes autophagy through 3 key mechanisms. The first mechanism is the MTR pathway inhibition via AMPK. The second mechanism is TFAB activation, and the third mechanism is the protein aggregate clearance.
So Trehalose clearly plays a powerful role inside living system. But Akmal, I'm curious, how is all of this actually being put to use in the real work, like in industries?
Okay, it was a great question, Amira. Trehalose unique properties have opened up a huge range of application, from the food we eat to the medicine we rely on, and even to cutting-edge conservation science. Let me break those down. Alright, do you guys ever wonder why some energy bars stay soft for 6 months on the shelf while homemade cookies go hard in 3 days? There's one reason, is the application of Trehalose in food preservation. Now I will explain to you guys about the food application of Trehalose. We have prevention of starch retrogradation, so it binds tightly with water molecules, prevent cooked starch from realigning and hardening over time, which preserve the texture and drastically extend the shelf life of baked good and starch-based food. Next, we have the protection against oxidation. Because it is non-reducing sugar, trehalose doesn't cause mild browning or caramelization, so it stabilizes delicate volatile flavor and prevent color loss in processed food and vegetable. So, how do vaccines survive a 48-hour trip across the country without refrigeration? That's where trehalose come in as a stabilizer in pharmaceutical formulation. So, we have two applications of trehalose in pharmaceutical. First, we have vaccine and mRNA stabilization. The sugar form hydrogen bond directly with nucleic acid and lipid nanoparticle deliver the vector. So this prevents structural degradation during freezing or freeze drying. They ensure the next generation therapeutics stay biologically stable without regress. So the next application we have induction of autophagy for neuroprotection. So trehalose has been shown to induce cellular autophagy through an mTOR independent pathway. So this specific capability make it a focus of clinical trial targeting neurodegenerative disease characterized by toxic protein misfolding.
So, that's Trehalose, a remarkably simple molecule with an extraordinary range of application.
From its biosynthesis inside living cells to its role protecting our health.
Right, all the way to preserving endangered species and revolutionizing vaccine storage. Trehalose is a protective sugar.